we just talked about the different kinds of amino acids. Now let's go about how they're broken down, or how they're destroyed. Now, as we just saw, not all amino acids are created equally. Martin Luther King Jr. would be very disappointed in amino acids. But like I just said, we just saw how they're different classified based on whether they're acidic, basic, neutral, essential, etc. Therefore, different amino acids go through different catabolic processes, many of which yield metabolic fuel in the form of pyruvate or acetyl-CoA, and then ultimately glucose and ketone bodies, but those pathways are not as high yield for your exam, and I'm here to get you the maximum points. I'm here to tell you what you should know for step 1. Now, all amino acid breakdown processes will produce ammonium, and if this ammonium isn't disposed of properly, it can cause disease. So how do we get rid of this ammonium here? Well, the body created a cycle, and that's going to be the urea cycle. Urea is going to be the non-toxic, disposable form of ammonia here. And it's going to be synthesized in the liver, and it's going to be excreted in the urine. Now, this accounts for about 90% of all nitrogen excreted in the urine. Alright, so let's move this off to the side and bring about the urea cycle. Now, let's talk briefly about this. The reactants for this urea cycle are going to include ammonium. Remember, we just said we broke down an amino acid and we created an ammonium. We need to get rid of it. Then also we've got a carbon dioxide, which in this case is going to be in the form of a bicarbonate ion. But remember, this is pretty much just carbon dioxide. Alright, so we have our reactants. Now, what are the products of the urea cycle? Well, hopefully you're thinking urea, because that's what we're aiming to create. But also notice how down here we have a fumarate. Now, where have we seen this fumarate before? If you're thinking back to this TCA cycle, you'd be correct. Notice how fumarate can feed back into the TCA cycle, thus everything within the body is going to be linked. Now, it's important to note that the urea cycle takes place in both the mitochondria of the cell and the cell cytoplasm. Do you remember what the rate-limiting step was for the urea cycle? Well, here we go. Let's create a mini board question. There are 4 enzymes here. There's carbamoyl phosphate synthetase 1, ornithine transcarbamylase, this one down here, and this one down here. There's 4 enzymes listed on this cycle. Which one's the rate-limiting step? Well, hopefully you're thinking this first one up here, the carbamoyl phosphate synthetase 1 enzyme. Now, this enzyme converts carbon dioxide and ammonium into carbamoyl phosphate. This should sound familiar since you heard about it at the beginning of the biochemistry chapter. Carbamoyl phosphate synthetase 2 creates carbamoyl phosphate in pyrimidine synthesis. Pyrimidine synth. Alright, so you've already talked about carbamoyl phosphate synthetase 2 enzyme, and you saw that in pyrimidine synthesis. Now we're going to talk about the carbamoyl phosphate synthetase 1 enzyme. Alright, it's also important to note that this process requires 2 ATP to occur to create that carbamoyl phosphate, which is going to feed into our urea cycle. We're going to need to invest a little bit of ATP in the process. The urea cycle ultimately yields blank, which is excreted in the urine, and blank, which can enter the citric acid cycle. You should knock this one out of the park, unless if you weren't listening, in which case you might struggle a little bit. Well, what do you think? It's going to create urea, and that urea is going to be excreted into the urine, which is how we get rid of the nitrogen from amino acid breakdown. And then it's also going to produce a fumarate, which can then feed back into the citric acid cycle, or the TCA cycle. Alright, as I mentioned before, ammonium is going to be toxic, and we need to get rid of it. So we need to convert it back to the urea so it can get excreted into the urine. However, the urea cycle only occurs in the liver, which means that ammonia must be transported to the liver first. How does the toxic ammonia that gets produced by the body, so let's say a muscle here, how does it get transported to the liver? That's going to involve alanine, which is shown here. In the muscle, the amine group that you're trying to dispose of is added to the alpha-ketoglutarate in the transamination reaction, which results in a glutamate. So by the way, this is the same alpha-ketoglutarate that we talked about in the TCA cycle. Everything's connected here. In a second transamination reaction, this amine group is transferred to pyruvate, which forms alanine. This is catalyzed by alanine aminotransferase, or ALT. Remember, it's that common ALT enzyme that you'll see on clinics. Now, alanine can then circulate through the blood and enter the liver. And once in the liver, the previous reaction is reversed, and the amine group is transferred from alanine to an alpha-ketoglutarate to form glutamate. And then in the final step, the ammonium from glutamate enters the urea cycle, and it's going to be used to create urea. So, get rid of that nitrogen. This also allows glutamate to cycle back to form alpha-ketoglutarate. Kind of a show that we need to balance out. We're going to make something and then convert it back, all in the hopes of transferring an ammonium group from the body to the liver where it can get excreted. Now, what might be some symptoms that you would see if you have too much ammonia? Or in other words, you're producing ammonia faster than you can get rid of it. Excessive ammonia is a serious medical condition because it can cause hepatic encephalopathy. Alright, so hepatic encephalopathy here is going to present with asterixis. These patients may have a tremor, slurring of speech, somnolence, vomiting, and blurry vision, and it can also be deadly due to the cerebral edema. It can be caused by liver disease. So the liver may not be breaking down the ammonia and be putting it into the urea form, or it can be due to a genetic deficiency of the urea cycle enzyme, which is going to prevent the urea cycle from working properly. So in either case here, ammonia will build up in the body, and it'll be transferred to alpha-ketoglutarate to form glutamate, as we talked about earlier. Now, this is going to deplete your supply of alpha-ketoglutarate. You're going to decrease your alpha-ketoglutarate. When the alpha-ketoglutarate supply is depleted, which important metabolic cycle is going to be inhibited? It's going to be your TCA cycle. Your TCA cycle will suffer if you have decreased alpha-ketoglutarate levels. So how are we going to treat this hyperammonemia? Well, the treatment of this condition is to limit the protein in the diet, because proteins are going to have our amino acids, which get broken down into ammonia. And then also we need to administer amino acid binders, such as benzoate or phenylbutyrate, which will increase the excretion of ammonia. You can also give lactulose to acidify the GI tract, which is going to trap ammonia so it can be excreted rather than absorbed. So let's take a quick look back at the urea cycle. Do you remember what the rate-limiting step of the urea cycle was? Alright, we already covered that. That's going to be our carbamoyl phosphate synthetase 1 step, and that's converting our ammonium and CO2 into a carbamoyl phosphate. Now, how does the body regulate this step? Well, N-acetyl glutamate is going to be a cofactor for this carbamoyl phosphate synthetase 1. So if N-acetyl glutamate is missing, the rate-limiting step for the urea cycle is going to be inhibited, and thus hyperammonemia may occur. So it's going to present the same as a carbamoyl phosphate synthetase 1 deficiency, but normal enzymes with an increase in ornithine, so an increase in ornithine can suggest an N-acetyl glutamate deficiency. Speaking of hereditary urea cycle enzyme deficiencies, we have another disease that we need to talk about, and that's going to be ornithine transcarbamylase deficiency. So here's our urea cycle, here's where the enzyme fits into the big picture. Ornithine transcarbamylase deficiency is the most common urea cycle disorder. Since it's the most common urea cycle disorder, it'll be probably most common on the USMLE. So let's learn about it. A lot of the different enzyme deficiencies that we've mentioned earlier are autosomal recessive. However, I want to note that this disease, OTC deficiency, is X-linked recessive. What does that mean? Well, let's go through a scenario. Can a male with ornithine transcarbamylase deficiency, which remember, X-recessive, can that father pass this disorder off to his child? His child. Terminology there. Could that mean son or daughter? Well, you have to play out the scenario for both. So let's go through it. Here's our father. He has ornithine transcarbamylase deficiency. And let's just say we have a normal mother. So, can he pass this off to his child? Yes, the father can pass this mutation on to his daughter, but not the disease. So he can pass the mutation on, but not the disease. So let's go through it. So the mother will always give one of her X's. Doesn't matter which X, they're both normal. So either child will have an X chromosome. The father can pass on the Y chromosome to create a son. Well, that Y is not mutated. Remember that mutation's on the X chromosome. So this son is normal. This son does not have the disease or the mutation. However, if he passes off his X chromosome, remember this is the one that is mutated. So the daughter will have a mutated X, but then they'll also have a normal X. Since it's X-linked recessive, this daughter will thus carry the mutation, but not the disease. So, remember, ornithine transcarbamylase is used to convert our ornithine into our citrulline. So remember how it plays into the urea cycle. It's going to also add a carbamoyl phosphate in the reaction. So if you don't have this enzyme, carbamoyl phosphate will build up. And if you don't have the ability to complete the urea cycle, you'll ultimately have ammonium building up, which is bad. Now, got a flash quiz here for you. A lethargic newborn with a low blood urea nitrogen is diagnosed with the most common urea cycle disorder. What substance will you find in his blood? Hopefully you're thinking orotic acid, which is going to be used for pyrimidine synthesis. This child has an ornithine transcarbamylase deficiency. Excessive amounts of carbamoyl phosphate are going to be building up within the body, which then gets converted into orotic acid instead of entering the urea cycle. Last slide, I briefly mentioned on how orotic acid can build up when ornithine transcarbamylase deficiency is present. Now, let's see why this is true. In the figure here, we see that we have ammonium that gets converted to carbamoyl phosphate. We already know that. Now, this carbamoyl phosphate has two options. So we have our ammonium, creates a carbamoyl phosphate. We have a couple options here. It can either enter the urea cycle, which we just talked about. So this is kind of our modified urea cycle. However, it can also be used to build pyrimidine nucleotides. Do you remember what the three pyrimidine nucleotides are? The three pyrimidine nucleotides are going to be your cytosine, thymine, and uracils. So, if you have a urea cycle deficiency, such as ornithine transcarbamylase deficiency, the carbamoyl phosphate will switch pathways, leading to an orotic acid buildup. Manifestations of this transcarbamylase deficiency include orotic acid in the blood and urine as well. You'll see decreased blood urea nitrogen, since urea can't be formed without this enzyme. And the symptoms of hyperammonemia, which I've already described in the previous slides, since ammonia cannot be converted to urea. You may also see high orotic acid in the urine and orotic aciduria, which is going to be caused by a defect in one of the enzymes that converts orotic acid to UMP. But do you remember how you can differentiate between these two? Orotic aciduria won't have hyperammonemia, since you can still break down ammonia just fine and excrete it in the urine.